So first of all, thanks to the organizers for giving me the opportunity to present our research. So I'm a scientist at, as you mentioned, UCSD and CDIPD. I fulfill some criteria required to be a scientist, like having a glass and baldness.
Yeah, well, not the bottom here.
Oh, no, yeah. Okay. So I'll talk about today two parasites, two amoebas specifically. One is called Entamoeba histolytica, which is the causative agent of Amebiasis, a diarrheal disease, and also it can go to the liver, causing amoebic liver abscess. And it is a global disease, and it leads to infection of 50 million people, leading to about 70,000 deaths each year, and about 500 million people are at risk. And there are two drugs required currently to treat this disease. The standard drug is called metronidazole, which has adverse effects, several adverse effects. Even you cannot drink alcohol while taking the drug, so that's also a thing for several people. And then there is a resistance concern with this drug. And remember, this is the only drug available to treat amebiasis, which was developed mid-50s. So for more than 50 years, we haven't come up with any backup drug in case there is a big resistance. Already we know from other parasites like Giardia, where this drug is used, there are several treatment failures. And also another pathogen called trichomoniasis, which is related to sexually transmitted diseases. So there are reported treatment failures with metronidazole on other organisms. Moreover, treatment with two drugs, metronidazole and paromomycin, takes 20 days, which causes compliance problems. So in order to develop a better drug, we need to come up with something where a single drug can treat this disease instead of taking two drugs and taking 20 days to treat one parasitic disease. Although it affects about 50 million people, it is a neglected disease. The reason is that there is mainly this disease is primarily of the poor, so there is less return for the pharmaceutical companies. So they are not interested in developing any new drug targeting this disease. That's why we came into the picture and work with this organism. In order to accelerate the drug development for this parasite, we first developed a screen. The challenge of developing the screen for this specific parasite was that it cannot survive in the oxygenated environment. Remember, this lives in the gut and where there is less oxygen. So it cannot survive in the high oxygenated environment, which is present in that microtiter plate. When we try to develop drug, we need to screen or investigate thousands of compounds which can be effective against the organism. And that's not possible doing in flasks. So that's why we wanted to move to the microtiter plate, where you can seed all those parasites in single well and then investigate thousands of compounds. But that well has a lot of oxygen, which can kill the parasite. So that was a big challenge. And there was also, secondly, no rapid robotic driven assay available. No readout was available before we started the project. So we overcome the challenge of oxygen. So we developed the screen which can be conducted in anaerobic condition, which basically mimics the amoeba's natural habitat. And then we developed a bioluminescence based assay which can be compatible with automation, so that thousands of compounds can be easily tested against this parasite. So the first screen which we performed, as Annette was mentioning, the repurposing thing, especially when it is a neglected tropical disease, so it makes a sense to try those compounds which are already FDA approved. So we went to a library which had about 1,000 compounds consisting of both FDA approved compounds as well as non-approved but bioactive compounds. The most potent compound we found, it's called Auranofin. that was about 10-fold more potent than the current drug metronidazole, which is being used. And that was in the culture system, in the plate. So you can see here the Auranofin 50% inhibition rate was like nanomolar range, whereas for metronidazole it was micromolar. And this drug has been in the clinical use for more than 25 years, since 1985, for the treatment of rheumatoid arthritis. So we wanted to follow up this one for a few reasons. First, this is FDA approved, has been in the clinical use for several years, so we know that this is quite safe. Secondly, this is orally available for those diseases which are mainly affecting poor. It should come up with something which is orally available instead of going through IV, all those things. But lastly, it is relatively cheap compared to other drugs. So in our follow-up animal studies, which we have, we compared Auranofin and metronidazole in the mouse model of amebic colitis. And a single dose, oral dose of 1 mg per kg per day for 7 days significantly reduced the number of parasites present in the amoeba in the mouse gut. Whereas same dose didn't cure mice with metronidazole. Same dose of metronidazole didn't cure the mice. Moreover, the 1 mg per kg per day for 7 days treatment of mice with Auranofin reduced the host detrimental inflammatory response as well, which was measured by myeloperoxidase level. So it was both anti-inflammatory as well as anti-parasitic. As I mentioned earlier, that in addition to amebic colitis, this can also go to the extraintestinal site and causes amebic liver abscess. So we have another animal model, which is in the hamster, where we developed liver abscess. So we tested Auranofin and metronidazole at 3 mg per kg per day for 7 days, and then we found that Auranofin significantly reduced the liver damage in hamster, whereas same dose of metronidazole did not have effect on liver damage progression. Then we wanted to know how this drug works in Entamoeba histolytica. So based on our transcriptomic studies, we hypothesized that thioredoxin reductase, which is an enzyme involved in redox detoxification, can be targeted by this drug in Entamoeba histolytica. And we confirmed that by biochemical studies, where we found that Auranofin clearly inhibits Entamoeba histolytica thioredoxin reductase. And inhibition of this enzyme makes the parasite more sensitive to oxygen-mediated killing. So when we did that experiment, we can find that in presence of Auranofin, there is accumulation of reactive oxygen species inside the parasite that is leading to the cell death. Now here in this picture, that green color depicts the accumulation of reactive oxygen species, which presages cell death. So based on this data, we went to the FDA. As I mentioned earlier, that the number of people get affected worldwide, it's a big number. But in the US, it is a rare disease. As Annette previously mentioned, several drugs first get approval for orphan drug status, and later it can be used for other diseases. So we went to the FDA armed with this data, and FDA was quite happy, and they gave us orphan drug status for Auranofin for the treatment of Amebiasis. Since the drug has been in use for 25 years and it's relatively cheap, so identifying this repurposed drug will significantly reduce the cost and development time. It takes so long for any drug to be developed, like on average 10 years and 1.5 billion, which we in the academia cannot afford. Since our discovery of Auranofin for the treatment of amebiasis, other people, our collaborators and we, also tested on multiple parasites to see whether this can be equally effective against those parasites. So we found that, as I mentioned earlier about giardiasis, it was equally active against giardia, which affect about 300 million people worldwide. Cryptosporidium, another diarrheal parasite, was also tested against with this compound and found that it was active against Cryptosporidium. Trichomonas, which I mentioned, causes sexually transmitted disease. We have recently got a grant working on this organism, and we found that Trichomonas is equally susceptible to Auranofin mediated killing. And that was a big number, also 174 million. We also published that Auranofin can work against filaria as well as onchocerca, which causes river blindness in Africa. So altogether, Auranofin was not only active on 50 million people suffering from amebiasis. Considering all those diseases, it can be effective against people, means 1 billion people suffering from several other parasitic diseases. So currently, we are in the phase 2 clinical trial that has been funded by the FDA in collaboration with Dr. Sharon Reed of UCSD. We have a clinical site in Bangladesh, so we are now conducting clinical trial for amebiasis and also giardiasis together in Bangladesh. And other clinical trial planning is happening. I know Gates Foundation is very interested doing the onchocerca project, onchocerca clinical trial, because they funded the onchocerca project. Moving from parasitic amoeba, I'll also talk about another amoeba, but that doesn't cause diarrhea or liver abscess. Instead, it goes to brain. And it is a rare disease in the US, and it is caused by free-living amoeba called Naegleria fowleri. And the disease is called primary Amebic Meningoencephalitis, or PAM. You may see this news every year during summer months in the US, and it's get well publicized. Every summer, few kids die from this rare infection. And the last case happened end of August, where this brain-eating amoeba, which is popularly known, popular name of Naegleria fowleri, killed a 14-year-old kid. This pathogen disproportionately affects children less than 13 years old. That's the average age, whatever we found from other data. Now, how it infects people. Usually when you go for swimming, especially kids during summer months, it enters through the nose and rapidly goes to the brain, and within 24 hours to 5 days, kids die. It is so aggressive, it totally kills the brain tissue. Now, that's why you can see some warning sign that do not allow water to enter the nose. But that's hard unless you wear a nose clip while swimming. This organism also survives in high temperature in freshwater lakes. It is thermophilic. It can tolerate up to 45 degrees centigrade temperature. There is an idea that with the global warming, we may see more cases in future. So now the goal is to find a good drug for this special disease. I'll say why there is a requirement for a good drug. If you look at the distribution of reported PAM cases, remember this is even not notifiable disease in the US. So over several years, there are 133 case reports of PAM, means almost 50 years. So it's not big number, it's really rare. But there are only 3 survivors in the US out of all those numbers. Globally, about a dozen survived only out of about 300 cases. But if you look at the map, you can see most of the cases you can see in the US, 111. Rest, only few numbers. So that's very unlikely because you are in the tropics. There is high temperature in water. How come they have less number? One reason is because of the poor diagnostics. This causes meningoencephalitis. Several times people may misdiagnose as a bacterial encephalitis. So in the other countries, we don't know whether several deaths were occurring due to this or bacterial meningitis. And so that cannot be the case, that only US has 111 cases, the rest of the world has so few. So most likely misdiagnosis, misreporting, and then not, even in the US it is not a notifiable disease, so we may have more. Mortality rate, it is almost universally fatal. Mortality rate is greater than 95%. And no optimum treatment is available. Only amphotericin B is the cornerstone of therapy. Now, over several years, 3 people who survived, they not only got amphotericin B, they got cocktail of drugs, several antifungals. So we don't know which one helped them to survive. The last case where the kids survived, they implemented hypothermia to reduce the body temperature, which was very unique. In addition to amphotericin B, they used another drug called miltefosine and then hypothermia. So we don't know whether any single thing affected the survival, whether hypothermia or miltefosine or combination of both, because another patient didn't survive with the same technique. So it is imperative to develop something to prevent these deaths, which can be rapidly progressed. Means you need fast-acting drugs because the disease, the amoeba progresses so fast. Within 24 hours to 7 days, the death takes place. So we, at that time, about 4 years back, we made a collaboration with a local biotech at San Francisco, which is called Asia Biotech. They had a compound called corifungin, and it is a broad-spectrum antifungal. Based on what we saw, that amphotericin B is an antifungal, and it's very likely some antifungals may have effect on this organism. Now, since it was a broad-spectrum antifungal, we wanted to test on the Naegleria and compare with amphotericin B how effective that antifungal is. And we found that it was equally potent. Amphotericin B EC50 was nanomolar range, and corifungin was equally active. But the advantage of corifungin was that it is highly water-soluble. Amphotericin B is a very toxic drug. It causes renal toxicity and has several adverse effects. So, but when this kind of disease is so fatal, you don't care, you want to take whatever kind of compound is available, right? Or the adverse effect is less important than survival. But the insolubility of amphotericin B leads to toxicity. So that's the most likely that is causing the toxicity. When corifungin was made soluble in water, it was hypothesized that it will be less toxic. Now, and in the later studies, we found that it was indeed less toxic. Even in rat, it can tolerate up to 250 mg per day. So at high dose, it can be tolerated. So obviously, when we found in the culture that it was very active against Naegleria, we wanted to test it in the animal model. So we have a mouse model where you can see that the parasite infection in the olfactory bulbs causes brain damage. You can see several trophozoites; those are the parasites, which are pointed by the arrowheads. And there are also inflammatory cells, mainly neutrophils, which are pointed by the arrows. And that was in the normal mice where they got Naegleria. So the gold standard amphotericin B, so we used 9 mg per kg per day amphotericin B for 10 days. It didn't improve. There was brain damage. Trophozoites or parasites are still present in the brain. So, and you have inflammatory cells around. So nothing happened in the amphotericin B treated cells at that dose. But when we tested corifungin at the same dose, we didn't see any parasites. What we had only inflammatory foci, because it caused inflammation, so you had some inflammatory foci, but no parasites were found. So clearly it had in vivo efficacy. And when we looked at the survival, all the mice survived, 100% survived throughout the observation period, whereas amphotericin B treated as well as control mice died within 10 to 12 days. Since it is a rare disease, we went to the FDA with this data, and we got orphan drug status for the treatment of Amebic Meningoencephalitis. So next steps involve phase 1 clinical trial, because we still need to show that this is safe in human. Phase 2 is not possible for Naegleria infection because the number is so low. So what FDA told that if you can show that it is safe for the human, then it will be distributed through compassionate ground. So they will give it to the CDC, and then the doctors can prescribe based on compassionate ground. In addition to this, I'm also involved finding other good compounds using target-based approach. We know that virulence, a major virulence factor is involved causing the brain damage. So I'm using several other inhibitors to test that whether that enzyme can be inhibited and whether that can also improve their survival. So with that, thanks for your attention, and I'll be happy to take any questions.